Hello, and a very warm welcome to this evening's lecture in remote as part of the Darwin Shrewsbury Festival 2021. Of course, with that theme in mind, today is a particularly auspicious day, being the 212th birthday of the great man himself. So, happy birthday, Charles. We have an absolute treat ahead of us. Before introducing our speaker, I'll just introduce myself and make a few essential announcements. My name is David Ray, and I'm the Head of Science Outreach at Shrewsbury School. It has been absolutely fantastic this year to be able to contribute to the Darwin Festival lineup with this evening's and a handful of other talks. If this is the first talk you've tuned in for this week, then I wholeheartedly recommend finding a bit of time to browse the archive section of the booking website, where you can catch up on fascinating talks from the last few days, which include a look at recent Galapagos finch population research by Professors Grant and Grant in Princeton, and an introduction to virus evolution and pandemics from Dr. Goldhill at Imperial College London, both absolutely cutting edge. I also need to tell you that this lecture is being recorded and that there will be opportunity for questions which will be answered at the end of the talk. Using the Q&A function, simply type your question and hit submit. So on to the main event of this evening. We're going to be given an insight into a little human brain evolution with a focus then on mental health and the applications of MRI and artificial intelligence. Our speaker, Dr. Kathryn Broadhouse, has recently moved to the UK from Australia, where she has headed research translating cutting-edge magnetic resonance imaging techniques to map and model mental health disorders and healthy aging. A physicist by training and holding a PhD in medical physics from Imperial, she's an Associate Senior Research Fellow at the University of Sydney and an Affiliate Senior Research Fellow for the University of the Sunshine Coast. Kathryn has recently taken up a new role of STEM Fellow here at Shrewsbury School, and over the course of just a few weeks, we've seen her passion for developing an awareness and love of STEM subjects in students and the community, and also that she is an exceptional communicator of science. Kathryn, we are very pleased to have you here this evening. Over to you.
Thanks, David. And thank you to the Darwin Shrewsbury Festival for allowing me to talk to you guys tonight about some of my past research. So tonight we are going to be traveling through quite a few different fields of science. There we go. We are going to see how we can combine all of these different fields of science to study the human brain, this sort of fascinating and complex organ that is the human brain. Now we're going to start off by just comparing the human brain to other animal brains. So in this image here, we've got 7 brains, and we start down the bottom with a fish brain. And then we move up into the amphibians, and then into the reptiles, and then into the bird kingdom where we have a goose brain here, and then into the mammals. So you can see we have a cat, and then a monkey, and finally a human brain. And kind of the first thing that pops out of you is that all of these brains look very different. They actually look like they're not actually the same organ. But from this sort of helpful little color key code here, you can see that we actually share all of the same components. And these components of our brain are sort of roughly in the same configuration. It's just their size and shape that is a bit different. And actually, you can tell a lot about the environment that the animal has evolved to survive in just by the shape of the different components of the brain. So it's a lot like the finch's beaks, I guess, in a way, because you can tell exactly the environment that this animal has evolved to survive in. And so as you can see, as we move up here into the mammals, so you can see that this yellow region here is actually the forebrain. And the forebrain starts to get really large in the mammals. And so much so that it starts to sort of fold in on itself. So that's because it's trying to pack in as many cells as it can so that we get a lot more computing power, a lot more processing power, because this is the part of the brain that gives us higher order functioning so we can problem solve and reasoning skills. So let's look at the human brain in more detail. So we can roughly divide the human brain into different regions. And these regions carry out different tasks. So just to orientate ourselves here, we've got the front of the head here. This would be the top of your head, and then the back of the head here, and the base of your neck would sort of come down here. So this pink region at the front is called the frontal lobe. And this is like the sort of control center of your brain. So it carries out all of your sort of higher order processing skills, your emotional processing, and it sort of controls your personality and our ability to communicate with others. Then this yellow region here, this is the temporal lobe. So this is the memory center of the brain. So this is in charge of creating and consolidating memories. And then these two colored stripes at the top, this is the motor cortex. And this part of the brain controls and plans and coordinates and carries out your voluntary movement. Then just behind that, we have the parietal lobe. And the parietal lobe processes sensory inputs from the outside world, predominantly to do with touch, taste, and temperature. And then at the back here, we have the occipital lobe, and that's the visual cortex. So this processes and interprets the world in which you see. So this is responsible for how you see the world, essentially. So what has this large forebrain actually given us? So if we're now going to move into psychology, and we're going to look at something called executive functions. So executive functions are higher order cognitive skills that control and coordinate lower abilities and behaviors. So they're things like cognitive flexibility, inhibitory control, and working memory. And so what I'm talking about when I say cognitive flexibility, that's sort of things like flexible thinking. So we're able to adjust our behavior and actions to unexpected changes that we meet throughout our day. Inhibitory control, so that's emotional control, so keeping our feelings and emotions in check, and thinking before we act. And then we've got working memory. So this is our ability to retain and call key pieces of information on the fly when we need them. And then combining these two skills, we have planning and prioritizing, organizing, so we can keep track of things physically and mentally. And then we also have self-monitoring skills. So we've got self-awareness, and we can assess how we're doing at any given moment. And then finally, we've got task initiation. So that means that we can stop procrastinating and start a task. So these executive functions are really important for us to just navigate through our daily lives. And they're also really important for us to be able to maintain our mental health and our well-being. So now we are going to look at the structure of the brain in a little bit more detail to see how we actually harness these executive functions that all the extra computing power that we have with our massive forebrains gives us. So this is an MR image of my brain. I scanned myself back in Australia before I came here. And I've just sliced my brain right down the middle. So this is my nose, it'll come off out the side here. My eye would be about here, and this is the front of my head. And again, this is my back of the head. And you can see that my brain is kind of nestled quite tightly in my skull here. And you can also see from the different types or the different colors, I guess the different gray scale on here, that my brain is predominantly made up of two types of tissue. So we've got the dark gray going around the edges here and some areas in the middle. And imaginatively, that is called gray matter. And gray matter is, essentially gray matter is the region of the brain that carries out all of your information processing. So your thinking part of your brain. And this is made up of neurons. And you can see here, this is just a schematic of a neuron. So you've got this neuron body, and then you've got the axon, which allows it to synapse onto other neurons. So it can pass the message between each other, and they can work out problems together. Now, the lighter gray tissue that's sort of connecting all of the gray matter is called white matter. And this is sort of like the neurocircuitry of the brain. So this is the wiring of the brain. So this is also made out of neurons, but these neurons are pretty special because they have myelinated axons, which sort of acts like insulation, which means that they're really good at passing information between each other. So they do really actually act like the wiring of the brain, and they connect all of the gray regions together. So the gray regions can communicate and talk to each other and carry out tasks. Now, this setup or structure of the gray and the white matter in the brain means that our brain can constantly control the underlying sort of flow of information. So our emotions, our feelings, our imagination. And it does so sort of like a network. But instead of a sort of social network where you have friends that are sending messages via social media, there are brain regions sending messages via neurons. Now, the way that these neurons, or rather the way that these brain regions work together, is quite similar to the way that our society works. So we have different sectors of society that carry out different tasks, such as the education sector, the health sector, and we're all working together using the infrastructure of our society to function successfully. So just as the infrastructure of our society is really important for our society to function, the underlying infrastructure of our brain is really important for this flow of information for our brain's network to function. And so what we see is that the underlying structure and biochemical makeup of the brain actually determines this successful flow of information throughout the brain. So for a successful brain network, which allows us to be cognitively and socially and emotionally healthy, we need a healthy structure of the brain. So what we often see in mental health disorders is that there is a disruption to the flow of information in this network. So there is a disruption and a change in the way that this network communicates. And we often see an underlying change to the brain structure, as you would imagine. And this means that we are then unable to cope with normal stresses in our life. So our productivity will go down and our emotional well-being and our mental health may suffer because of it. So neuroscientists, or scientists within the field of neuroscience, so neuroimaging, we actually seek to examine this brain's network, so our structural and our functional network, to understand more about mental health disorders, to understand more about how the brain functions in health and how it functions in disease, and compare the two. So I guess mental health has kind of been brought to the forefront this year with COVID, so we're probably all quite familiar with these stats, but just to kind of run through again why it's so important to keep researching mental health. So about 1 in 4 Britons will experience a mental illness in their lifetime, and mental health accounts for about 27% of total years lost due to disability globally. A major depression is the second leading cause of disability worldwide. And also the prevalence of mental health disorders is greatest among 18 to 20-year-olds. Okay, so how do we actually go about studying mental health disorders? Well, we can use a lot of imaging modalities, but I've throughout my career used MRI, and that's because MRI is quite versatile, or rather very versatile. So what we can do is we can do a structural scan here, which is very similar to the image I showed you previously. And we can scan all the way through the brain. And you can see that this is really good at showing us the grey and the white matter distribution in the brain. So we can start to measure the quantities and the distribution of those two tissue types. We can also run the scanner in a slightly different way. So I've scanned this same person here, this is their same brain, but I've scanned it in a slightly different way. So I'm just looking at their white matter. So I've just pulled out their white matter fiber bundles. So I'm just looking at the wiring of the brain. And you can really see that it does actually look like a load of fiber optic cabling. And then what we can also do is we can run a type of scan called functional imaging. So this was a video, I didn't take this video. This video was published in PNAS in 2014 by Andrew Zaleski. So if you want to read more about this study, you can visit this website here. So essentially what we do is we measure the brain activation in the scanner while people are at rest. And what they've done is they've got a load of different brains, and they've just got them to lie in the scanner at rest, and they've measured their brain activity. And then this is denoted here by the color bar. So the higher, the sort of more red the color, the more brain activation. And what you can see is that all of the brain regions are kind of lighting up together and then switching off together. And that means that they're communicating together. So those brain regions are working together and constantly communicating. And you can actually see the flow of information through our brain's network using this scan. So with the MRI, we have the ability to look at the structural configuration of our brain, look at the structural connectivity of our brain, and the functional connectivity of our brain. So we can analyze all aspects of our brain's network. Okay, now, the fact that we can look at the brain as a network is really, really useful, because we've sort of, I guess, mirrored the internal organization of our brain in our everyday lives. So we are all a part of social networks, either on or offline, or both. And we use networks all the time to just survive and get by. So we have transport networks and trade networks. So that means that we have invented an entire branch of mathematics just to describe and analyze networks. And this is known as graph theory. So how do we do this? So this is a very, very simple brain here. And I've got my brain regions in blue, and I've got my communication pathways or my structural connectivity here in red. And what I can do is I can simply sum together all of the nodes. So how many nodes, and that's the mathematical definition for the brain regions. So how many nodes do I have in my brain? And the answer is 7. Once again, this is obviously a very simplified version of a brain, but just to show you what we do. And then we can also count the number of communication pathways. So the mathematical name for this is the number of edges, and that's also 7. And then using these numbers, we can start to look at network connectivity. So you can imagine that if we've got a brain that's really connected, or the brain regions are really connected together, then the number of communication pathways per brain region is going to go up. So that's another thing that we can assign a value to, we can quantify. We can also look at the average path length. So that will tell us about how the flow of information is going to travel around the brain. If information has to travel a long way to get to regions that are far apart, then that's probably not very efficient. And that's the case where we have a lot of small connections between nearest neighbours and not very many long distance connections in our network. So on the same hand, we can also look at network efficiency. So how quickly can a message, I guess, travel from one node, so one brain region, to another? And we can put a numerical value on it as well, because we know the number of nodes, the number of brain regions, and the number of edges. We can also pick out really important hubs in our network, so really important brain regions that have a lot of connections and communications to other brain regions. And that's sort of similar to sort of an airport hub. So Heathrow would be a really important airport hub, or rather it was before COVID, where we have a lot of routes flying into that. So we can define, let's say, a hub in our brain as a brain region that has a lot of connections. So I'm just going to say that this is our hub, so we have one. So you can quickly see, this is already very simplified, but you can quickly see that if we do this for different brains, we now have a way of being able to mathematically compare the networks. We have a way of being able to analyze them with statistics. So we can take an average network, brain network, for healthy controls. So in research, when we design a study, we'll have healthy controls, so they are the controls, the healthy participants, and then we have the disease participants, so the disorder that we're trying to study. And we can compare the average network, and what we actually see is structural and significant mathematical differences in their networks and the way that they communicate. So one network that I want to talk a little bit more about is the hippocampal prefrontal cortex loop. So don't worry about remembering that name. That's just kind of a long-winded name for the regions in this loop. So what I've done is I have taken my MRI scan and I've just done a surface rendering here of my frontal lobe. So that's in red. And so remember, that is the control center of the brain. So that's kind of carries out your higher order processing and your problem solving. And then we've got the hippocampus here. So the hippocampus is in your temporal lobe. So it's actually the memory center of the brain. We actually have two, one on either side of our brains, but for simplicity, I've just shown the left side of my brain. And then I've also surface rendered my amygdala, which is my emotional processing center of the brain. Now, what I want you guys to do is just imagine a scenario where we need to learn something new. So we've gone for a walk and we've got ourselves lost and we don't recognize where we are and we're in a bit of a panic. And we're trying to work out where we are. And the task is that we've got to work out where we are, try and orientate ourselves so that we can get back home safely. So what's going to happen first is that your amygdala is going to have an emotional response to this strange new environment. You're going to start to panic. And it's going to start communicating, sending messages to your frontal lobe to say you need to process this emotional response. Now, your frontal lobe is going to make a decision using all of the sensory input from your 5 senses, whether or not we are in any danger, whether we need to run away and get to safety, or whether we can actually stop and think about our surroundings and work out this problem a bit more. So now once we've worked out that there isn't a problem, it's going to suppress that message from the amygdala. That's going to free up a lot of your cognitive processing so that you can actually start to use your executive function. So we've already had inhibitory control here, and now we're going to move into our cognitive flexibility. So we're going to start to use this big part of the brain to work out how to get home. And now our frontal lobe is going to start talking to our hippocampus, which is the memory center of the brain. And it's going to start to recall old memories, things that may help us in our predicament. So do we remember how to find north and south from the position of the sun, let's say? So as we're recalling old memories and using that to learn about our new environment, we're also laying down new memories. So we're learning new things. So that's working memory. And then as this is all going on, our hippocampus is talking to our amygdala, and our amygdala is assigning a positive or negative association with this experience. So that's really important, because that essentially gives you motivation to carry out task again. So if you're enjoying something and you assign positive association with that experience, then you're more likely to do that again. And if it's a bad experience, then you don't really want to revisit that memory. And so as you can see, this is sort of going on all the time as we're trying to figure out where we are and learn about our new surroundings. So you sort of get a feedback loop. And if we keep doing this more and more, so let's say we keep revisiting, we find our way home, and then we keep going back there and revisiting that place and using the same or activating the same neurocircuitry again and again, what we'll find is that this neurocircuitry will change and strengthen so that it becomes more efficient. So this is kind of like the neurobiological underpinnings of the phrase practice makes perfect. So when we, let's say, learn a new instrument or something, or a new language, it's really difficult at the beginning. And that's because our brain is sort of working out exactly the neural circuitry it needs to perform this task. Once you've done it for a year, you sort of, it's become sort of like a habit. So you end up changing this circuitry so it becomes more efficient, so that your brain uses less energy to perform the same task. And this is known as neuroplasticity. So we get kind of improved learning with more neuroplasticity. So this isn't just me kind of colouring in my brain. This is a real thing that we've observed in the research. So this was a study carried out by Martin Dressler. And what they did was they took a load of memory athletes. And so memory athletes are people who remember long lists of things, and they compete at an international level. So they all meet up and they recite long lists of things. So they might recite pi to sort of the 1000th decimal place or something. So they have extraordinary memory. And what they did was they put them in the scanner and compared them to controls. And they looked at their network, and they saw that these memory athletes had significantly increased connectivity between our frontal lobe and our temporal lobe, between our sort of control panel of our brain and the memory center of our brain. So that's kind of similar to what we've just seen with my coloured in brain earlier. But then, so what they thought was, well, is this just something unique to their brain? Have these people got something special about them that means that their circuitry is changed from birth? So they took the controls and they got them to train using the same techniques that the athletes were carrying out before they trained for a competition. And they found that the controls brain network shifted to be more like the athletes after training. So that's a kind of real life example of positive neuroplasticity in action. And that your brain is sort of it's not a fixed thing. You can constantly learn new things and sort of evolve your brain on a sort of micro timescale to suit you and kind of improve, I guess, your well-being. Okay, so now we are going to look at the impact of stress on the brain. So our brains have a sort of love-hate relationship with stress. I guess we're also creatures of the universe that obey the laws of physics. So when we're left to our own devices, we will sort of gravitate back to the lowest form of energy. So we need a bit of stress to kind of motivate us and up our performance level here. But too much stress and it can become quite toxic and paralyzing. So I'm just going to go back to my coloured brain again. So back to my hippocampal prefrontal loop. And we're going to imagine now that we have to prepare for a really important meeting, let's say. Or we have to revise for an exam and we've left everything to the last minute, so we're cramming. So we're really, really stressed. What happens now is that your amygdala's had this emotional response, and because it's really, really stressful and it's had a very big emotional response to this situation, it's almost sort of shouting at the prefrontal lobe, at the frontal lobe. And so this means that there's less cognitive processing power to carry out all of your problem solving. And the response, inhibitory control response, is dampened. So it's almost like the amygdala is screaming at your frontal lobe. And this has a knock-on effect, because as your frontal lobe is trying to process this huge emotional response to this situation, there's it's less likely and it's less efficient to be able to communicate with the hippocampus. So the connectivity and the communication flow between your frontal lobe and your hippocampus is impaired. So this means that you're not able to recall information as well. So we all know that feeling when we're trying to cram for something, we actually become really forgetful. So when we're stressed, we become really forgetful. And this has a knock-on effect as well. So your ability to actually lay down and consolidate new memories is also impaired. So it's actually when you're really stressed, the memories that you lay down when you're really stressed aren't actually very stable or reliable. And then if we continue around this loop, we're also having the amygdala here communicating with the hippocampus. And because it's a stressful situation, it's actually laying down a negative association with this experience. And then this has a knock-on effect here, and you can see that communication within this entire network is impaired or altered. Okay, so that's sort of a one-off situation, and we can all find ourselves in those with those sort of situations where we've left everything to the last minute. But periods of prolonged stress can actually lead to negative neuroplasticity. So they can change the brain's network and function in a way that's not very beneficial to our mental health or our productivity. So, and we can actually see this in adult studies where we've shown that the functional connectivity in their brain is altered in adults who have chronic stress and who also have major depressive disorders and anxiety. Okay, so now this is a study that looks at the onset of mental health disorders. So looking at the age of onset of different mental health disorders. And what we can see is that even though the prevalence is highest in years 18 to 24, the actual onset of most diseases is in adolescence. So 50% of mental illnesses begin before the age of 14. So there is now a lot of studies around the world that are looking at scanning, longitudinal studies, I should say, that are looking at scanning the adolescent brain, so teenage brains, and tracking them through their sort of adolescent years to try and understand more about the developing brain and understand more about the onset of mental health disorders. And so I was involved a few years ago in the longitudinal adolescent brain study on the Sunshine Coast. And that aimed to take 12-year-olds and scan them multiple times over about 5 years. So we took a preliminary data set when we'd finished the first year, and we just looked to see if we could find any correlation between brain structure and psychological distress scores. And what we found was that even though they were a lot younger than most people who than 14, so before sort of the onset of most mental health disorders, we could already see that there was a significant negative correlation between their hippocampal volume, so their memory center, and the higher distress scores here. So it's really early days and we don't know what that means. We don't know whether that might be a biomarker for disease. So a biomarker is a marker in the brain that we can use then to predict the onset of disease. So we have to keep following these kids over the 5-year trial period to work out whether small hippocampal size can actually predict the onset of certain disorders and diseases. Okay, so some of these examples that I've shown you, or most of them rather, are from the sort of research side of neuroimaging. And there's also the clinical side of neuroimaging. So the clinical side, you're having a one-off scan, and a radiologist will will diagnose if you have a problem. So both of these sort of modalities of using MRI, I guess, are very time consuming. They take a lot of man hours, a lot of computer processing hours, and they require humans and experts to be able to get it right 100% of the time. And quite often, obviously, well, not quite often, but sometimes just being humans, we miss things. Or whether in research, if we don't think that we need to look, by definition, we won't find things. So this is why we are looking to artificial intelligence to aid in this sort of accuracy and the detection of diseases, so the accuracy of diagnoses. Now, most people think of artificial intelligence as sort of the traditional rule-based AI. So this is where we have the algorithm behind the AI will be, they'll have loads of rules and it'll be based on rules and logic. So you may remember back in 1997, there was a rule-based AI called Deep Blue, which was programmed to compete in chess tournaments and beat all of the world champion chess players. But the problem with this is that when you pick it up and move it to, say, a game of checkers, it will have no idea what to do. So there is no flexibility in this algorithm or this code, and there's no flexibility to learn. So what we are looking at is, in the neuroimaging field, is using machine learning AI to help us. So machine learning AI is the opposite. So it has very few rules, and it can actually learn. And the way it learns is because the algorithm behind machine learning, artificial intelligence, is built on our model of how the brain works. So we call the algorithm behind machine learning AI neural networks. So we use neural networks to essentially learn the mathematical relationship between our input data and our output data. So now that kind of sounds a little bit vague and abstract. So I'm going to talk a little bit about how that actually works. So what we're going to do, imagine we've got a totally brand new neural network here. So it knows nothing. It's a completely new brain, if you like. And what we want it to do is to learn the mathematical relationship between the physical features of finches, so beak shape and feather color. And we want it to be able to learn how to classify from those two types of information, how to classify the different types of finch species, in particular these two. So we're going to get a massive training data set. So imagine we've gone to the Galapagos Islands and we've taken a load of photos of these two types of finches, and then we've got an expert to go through and classify which one of these is which. So we already know the answer. And then we're going to feed the images into our brain, and so our neural network. Now, these nodes in the middle are like brain regions. And so what they're going to do is they are going to make a wild stab in the dark at the first pass, and they're going to say, I think that feather color is really important to defining the species and beak shape not so much. So then I'm going to put a weight on my connections to the input data. And the other brain regions, because they process data in a slightly different way, much like our brain does, they're going to give slightly different weights. And again, it's a total guess, starting from scratch. And again, because they process data in a slightly different way, like our brain regions, they have different biases. And then again, as the connectivity between them and the output, they're also going to add weights. And then we're going to feed in our first image. And it's going to go through the model, and we're going to end up with an answer. And our brain is going to say that given these weights that we've decided to give our connectivity, so the strength of our connectivities between our input and our brain regions, we are 99% sure that this guy here is a ground finch. And obviously, because this is a training set, we know that that's incorrect. So we tell it it's incorrect, and then our neural network goes, okay, well, I'm going to adjust all of my weights and all of my bias. And it's going to go round and round and round, and it's going to do this with hundreds and hundreds of images that we already know the answer to in our training set, until it gets to a point where it can accurately predict or accurately characterize and define each image until we get it right. And then what it's done is it has learned on its own the mathematical relationship between beak shape and feather color and how that determines the species. So you can see that if we obviously this is a very simplified version of this, but this has quite a lot of power because we can use it with imaging data. So instead of our feather color and our beak shape, we can put in as our input data the physical and functional structure of the brain. So we can put in the functional connectivity information, let's say. We can also put in the structural information of our participants. And then if we train it on enough data, it will learn the mathematical relationship between these input features, so between our functional network, our brain network, and between the structure, and it will learn how to diagnose different diseases. And obviously this is far greater than any human brain could do. This is obviously going to take thousands of computers working together to work this out. So it has the potential to be a lot more powerful than the human brain. But it also has the potential to not be as powerful as the human brain. So the neural networks in machine learning AI are only as good as our training data. So if you imagine that we've gone on holiday now, we've set up our neural network traits, it's learned the algorithm and we're happy, we've passed the training stage, and now we're we need to use it on real data so that we can classify images and not have someone spend ages and hours go through and tell us what species it is. We just want to go to the Galapagos Islands and take a load of photos of finches. And we end up taking a picture of this guy and we pass it through our neural network. And it will, because it's only trained on these two images, it will always give you an answer that's either this guy or this guy. It doesn't know that this is not either of them. So it will say, oh yeah, this is the green warbler finch. The problem is that if we have that scenario in a clinical setting or in a research setting, we could misdiagnose someone. So if we come up with a false positive and we say, yep, you're healthy, purely because we haven't had a large enough training data set with all of the different diseases and disorders that someone could have, and we miss them, then that's a huge problem. And so if we become too reliant on our machine learning AI, we could actually misdiagnose people. So you can see that there's obviously a lot of potential for machine learning, but we are not at the stage yet where we can sort of mass roll out this technology and use it in the clinical and the research setting. And that is because our brains are amazing, really. They kind of sit in between these two. So although machine learning is based on how our brain, or how we think our brain learns, our brain actually sits between the two. So it has this kind of inbuilt ability of rules and logic to work out when an answer is actually garbage. So we can learn from a specific environment, and we can pick up what we've learned and use it on a completely different challenge. So we can apply what we've learned to completely different scenarios. And so machine learning just cannot do that. So our brain is actually quite incredible, really. And it does this just on 2,000 calories a day and 2 liters of water, whereas machine learning AI needs a sort of warehouse full of computers. So we are still learning a lot about our brain from AI, because when machine learning AI sort of goes wrong, it actually highlights how amazing our own brains are and how they don't go wrong most of the time. So I guess I'm going to finish up by this slide, which is, I guess I'd sort of be remiss if I gave a talk on mainly mental health and then didn't talk about COVID in this year. So it's obviously been an extremely difficult year for everyone around the world. But I kind of like to try and find the silver lining. And I think that this has actually been a year where most people have been extremely aware and open about their mental health. So there's never been a time in our society anywhere where we are so open about talking about our own mental health. And we're also really aware of the importance of exercise and our social networks and connections with our friends and loved ones. And so I guess over the year, most of us have found that we are actually changing our behaviors to make those more important and predominant and dominant in our lives, which means that we are changing our behavior. And if we're changing our behavior, then we're setting up neuro changes in our brain so that we can add more resilience and hopefully tackle the problems that present us in the future. So with that, I would like to end the talk, and thank you very much for listening. And I'm looking forward to any questions that you may have.
Wow. Well, thank you very much, Kathryn. That was truly fascinating. And although it may seem a little bit strange, I'm sure the viewers at home will join me in a kind of personal round of applause, which we know we can't hear, but I feel like it's the right thing to do. And that was really great. Thank you. We are going to give over the rest of our time to a question and answer session. We do have a few questions already starting to appear in the feed here. I'm just going to say we're going to aim to finish at 8:30, just so people can get on with their sort of evening plans. So really sorry if we don't get through every question, but we'll kick off with one from Mr. Andy Coolermans. Great to see a familiar name there. He has asked, does neuroplasticity get disabled or slightly less good with age? Is it possible to keep your neuroplasticity kind of going strong?
Yes. Oh, I'm so glad someone asked that. So glad Andy asked that. Yes. So this sort of notion that old dogs can't learn new tricks is not true. So your brain network can change throughout your life. I actually spent a lot of time before working on mental health, working in healthy brain aging and dementia. And so we were looking at lifestyle interventions to prevent the onset or push back the onset of dementia and mild cognitive impairment. And what we found was that exercise is actually one of the best ways of keeping mentally healthy. And we actually found, we took about 100 people with mild cognitive impairment, and we got them to do a 6-month training regime. Some of them did exercise and some of them did cognitive training. And what we found was that those who did exercise, their hippocampus, so the memory center of the brain, actually, so I guess I should start by saying that in age, your brain does shrink, and that's kind of normal. But when you get dementia and mild cognitive impairment, that sort of shrinking and degeneration of the brain increases. So you lose brain volume at a faster rate. And one of the first areas to suffer is the hippocampus. And what we saw was that there was a decline in this rate of shrinkage, I guess, with exercise. So they were able to sort of maintain more of their volume of their hippocampus by purely just doing exercise. And that actually continued a year after they had finished with their training program. So they could see the effects not only structurally in their brain, but also when we did a load of cognitive testing, we also saw the effects of their in their executive functioning as well. So yeah, it's never too late to start.
Great, thank you. We got one from Jane McKenzie, who asks, how has neuroimaging helped to understand the origins of addiction?
That's a really, really good question and a really tough one to answer. We still don't understand the origins of addiction. We can look at the changes, I guess, in the brain networks with people who have had problems with addiction in the past. However, we aren't at the stage yet where we've scanned enough people, because obviously to be able to pull out the onset of addiction, you'd need to scan a load of people and follow them throughout their life, and then see how their brain changes. So that then we can work backwards and see, well, was there anything in their scan before they had problems with addiction that may predict or indicate that they would be more likely or have a sort of disposition to become addicted to substances. So we just don't have those studies out there at the moment. So it's kind of quite a difficult situation, I guess. But people are obviously starting at the other end, where they're looking at the brain changes with addiction and trying to work backwards. So it's probably an area of research that's going to expand. And I guess with AI as well, they'll be able to take all of that data in and process it in a much more meaningful way.
Brilliant, thank you. A light-hearted one, and one for maybe someone thinking about the future. What was university like doing neuroimaging?
So actually I did just straight physics at university, and then I did neuroimaging as my PhD. But it was great. So university was great. I loved it. And I guess I loved my PhD as well. It was quite fun. It was quite, I actually did my PhD looking at the cardiovascular function of premature babies. So completely different organ and I guess different end age of the age spectrum to dementia. But you saw I was just awestruck by, I guess, the NHS. And because I got to work closely in a hospital in a neonatal intensive care unit, and it just blew me away. The struggles and that they go through every day was just incredible to watch them sort of meet that challenge every day. So yeah, it was really worthwhile doing.
A change of direction now into kind of more mathematical regions. We got one from Mr. Ian Payne, who is asking how stable the machine learning algorithm is, and with a lot of connections, is it potentially chaotic? Now, I'm a lay person when it comes to maths, but I know that chaos kind of has a significance, doesn't it?
Yeah. So it's kind of, we run into the problem where we can sort of overfit our data. So it all comes back to the training data set. So we want enough variability in our training data set so that neural network can learn appropriately. So if you have, I guess, too narrow a training data set, then you run into the problem that I described earlier. And if you sort of add too many layers to your neural network, then you're going to get overfitting, which is also a problem, because that leads you to believe that your accuracy is really high, when actual fact it's not. And so when you then put in real data, it will then come up with false positives as well. So there is, you definitely have to know what you're doing when you're first starting out with your neural networks, because it's like, it gives you quite a lot of promises, really, doesn't it? So it says, well, I can work out the relationship between X and Y for you. So in that respect, it's quite tempting for people who don't know much about their inputs and the outputs to just use it, because it's like a black box. You can just, and you always get an answer. And that kind of is where problems arise, and where you get really funny. I don't know if you guys have seen this on the internet, but there's sort of a machine learning AI algorithms that have produced some really crazy answers when they've combined different pictures together, and it's classified an avocado as a dog or something, which, I mean, that's sort of the funny end of it. But if we're doing it in medical imaging, it can be a real problem.
Well, we have a very related question, really, from Mark and Janet, who ask, how do you really know when you've provided sufficient data set options to make good selections?
Yeah, that is also a really good question. And it relies on the people who are putting the data in to be experts in the field. So the machine learning algorithms that a lot of the imaging vendors, so Siemens and Philips, are developing up at the moment, they're obviously consulting a lot of experts in the field of computer science, of neuroscience, of biology, and of medicine, so that they know that the answer is correct. Okay.
We are sort of going back to Andy's question a while ago about maintaining plasticity and brain health. Is it physical or mental exercise which was the best? I think you touched on this earlier.
Yeah, so in our research, it was the physical exercise. So exercise is, if it was a pill, it'd be like the wonder pill that every pharmaceutical company would want to own the rights to. But exercise is amazing. It has shown to be efficacious across not only mental health disorders, but neurodegenerative disorders, so at all sort of age spectrums. So I can only encourage people to sort of get out and exercise more, really. Yeah. So I guess cognitive training is, it's good, and obviously it kind of led to that flourish of those brain training apps. But it only improves your cognitive function in the task, in the domain that you're training. So if you go and do a load of brain training and the app is just memory, then your memory is going to improve, but it doesn't really have a lot of transfer. Whereas exercise, if you do exercise, it seems to improve all of your executive functions. And we don't fully understand the sort of pathway and the mechanism why that's the case. But yeah, I'd say get out and run.
Makes an easy solution, though. Just
yeah,
it will certainly be better. Yeah. Also on the topic of neuroplasticity, Sean has asked, how does CBT and mental attitude help with mental health, and can that aid neuronal plasticity between brain regions that may already be suffering mental health disorders?
Yeah, so CBT, you mean cognitive brain training, is that correct?
I guess so, yeah. Sounds sensible.
Yeah. So mindset, I'm glad you mentioned mindset. That is a really important question. I guess I've worked with one of my colleagues back in Australia. He looked at mindfulness as an intervention to prevent cognitive decline in aging. And mindfulness is obviously learning how to interrupt those negative thoughts. So learning how to stop that negative fixed mindset and start to expand and have a flexible mindset. And what he found was that those people that carried on with the mindfulness did really well. So their mental health improved, so across all measures, and they, I guess their cognitive function improved as well. So it is about mindset. Everything is about mindset, I guess. If you've already decided that you can't do something, then what you're doing is you're setting up that feedback loop in your brain. You're setting up a negative feedback loop because you've already assigned a negative emotional response to whatever task you're about to approach. And so you've already kind of set yourself up really to not get the best neuroplasticity that you can. So I would say that approaching everything with an open mindset is always a good thing to do.
Sure. And kind of again a follow-up, I guess, for maintaining health, good mental health. Gwyn asks, what about sleep? Where does that come into all of this?
Yes, sleep is it's amazing, really. So sleep, when I was down in Sydney, we were looking at the potential of sleep as well to impact on memory consolidation, particularly in the aging population. And so what we found was that when we combined sleep and improved sleep techniques, or rather what they found, because I was only part of the imaging of that study, and memory training, the people who had better sleep actually had better memory consolidation. So sleep sort of acts as a regenerator of your brain. It sort of cleans out your brain, and it also kind of heals your brain overnight so that you're able to not only consolidate memories in your sleep, but then if you get a good night's sleep, you're able to, your ability to consolidate memories while you're awake also improves. So it has a sort of double whammy effect. So it is a big area of research at the moment that people are looking into, linked with mental health and also healthy aging.
Thank you. Well, I'm going to steer things back towards the idea of AI for a moment.
Mm-hmm.
To what extent is a neural network a physical thing, or is it purely code?
Purely code, yeah.
Okay, so the big warehouse is full of kit. That's just processing power.
Yeah. So obviously, you can imagine I only had 3 nodes in my neural network and only 1 layer, but actually we have obviously loads of nodes and loads of layers, and so that there are loads of connections, connectivity strengths, and biases that we're constantly changing to work out that mathematical relationship between input and output, and that requires a huge warehouse of computers. But the actual neural network itself is just an algorithm; it's just a code.
Okay, so sort of following on, Scarlett Gibson asks, will the role of AI become more useful, do you think, in the near or far future?
Yeah, I think for things like this, for applications in neuroimaging and in the clinical space, yes. I always have a bit of a funny feeling with AI, just because with mental health research, you learn that the most sort of important thing, I guess, is to stay socially connected. Is to be connected in your social network. So loneliness is really bad for onset of mental health disorders, as we all kind of can feel. We all know that already. And I feel that AI removes us once again from people. And so I think if we're sensible and we can keep it to just be something that we use as an aid to help us improve our, I guess I'm just talking in the imaging space now. If we just use it as an aid to help us diagnose people and investigate the brain more, I think that's really useful. But there is talk about using machine learning and artificial intelligence to actually talk to patients who have mental health disorders, so that they always have someone there that can talk to them. I don't think you can really replace people with AI. I think that just kind of adds to the problem, particularly when we're talking about mental health.
Okay, I think probably in an imaging sense, how do you go about detecting depression in a human brain?
So currently, the only way that we can diagnose mental health disorders is with a battery of psychological tests. So we only use MRI just to, in the research setting, to investigate the functional and structural changes in the brain. However, with research studies, what we found is that there are significant changes in the communication between our frontal lobe and other regions of the brain. And we actually have a network in our brain called the default mode network. And this network is active when we're sort of daydreaming and when we're self-ruminating. And what we find in people who have depression is that that network is turned on. It has stronger connectivity than most. So what we've sort of hypothesized is that people with depression end up getting stuck in this default mode network loop where they're sort of self-ruminating, and therefore it's harder for them to actually engage with the outside world and see themselves in the outside world. And so they're sort of stuck self-ruminating in their own minds, which can be quite a sort of negative spiral to get into.
You mentioned in your talk a longitudinal study over 5 years, which I think was looking at the hippocampus size and kind of stress scores from questionnaires of some sort. Mm-hmm. How far along is that research, and what conclusions have been drawn from it, if any, at this stage?
Yeah, so that study started in 2018, and they aim to recruit 500 12-year-olds and then obviously scan them over until they were 17. So there's sort of a rolling recruitment, because we obviously couldn't get 500 12-year-olds all through the door at once. So they are now sort of 2 years into their study. And I think they have scanned, when I left Australia, they'd scanned about 100. So they've got 100 through, and they're all sort of coming back for their first year and their year and a half scans. So we have looked at the preliminary data looking at hippocampal structure. And I know that one of my old PhD students, who's currently still working there, is looking at sleep and the correlation between sleep and the white matter connectivity in the brain. And he's found some really interesting findings about how sleep can alter the white matter connectivity, even in these sort of young 12-year-olds before they've gone through puberty and adolescence. So hopefully when they complete and we've gone through the full 5 years, there'll be some really interesting findings.
Wow, yeah. We've got one from Kate, who's asked, how do mental disorders affect different aspects of the brain? And for example, does autism affect the emotional and communication part of the brain, or how does it?
That's a good question. So I haven't ever researched autism, so I wouldn't know specifically. But I mean, with most mental health disorders, they're actually trying to rethink the way that they diagnose mental health disorders, because the traditional method is sort of based on psychological tests that were devised sort of 40 years ago. So they're now sort of shifting into this idea that we can maybe diagnose mental health disorders using the brain network. So it's almost like a fingerprint of the brain. And when you look at your brain functional connectivity network, and can we use that to then actually look at and diagnose these different disorders? Because what we find is there is a lot of overlap between someone with, say, depression and anxiety, and there's a lot of comorbidities as well. So someone with autism is more likely to have other mental health disorders as well. So it's quite a complicated thing to answer, I think.
Yeah, I can imagine. Ian Payne is wondering, does the brain have pre-programmed modules, or is everything learned?
No, we do have pre-programmed networks. So the default network is one of them. There's about 7 that we've identified in the brain, and there's sort of more popping up. So we have these kind of built-in networks that our brain relies on all the time. And then when we're kids, we have white matter fibers and white matter connections all over the brain, going everywhere. And I've got a really, really packed brain. Actually, I should have got an image, but when you compare a child's brain to even a young adult, it's actually quite amazing how much it's shrunk already. And it's good shrinking. It's not a problem. But what happens? It's normal. What happens is that when we go through puberty and adolescence, our brain starts to prune itself. Because it's trying to make that brain network more efficient, and it uses the experiences during adolescence to work out which brain networks it needs and which it doesn't, and so which connections it can strengthen and which connections it can sort of discard or just keep on the back burner. And so that is one of the reasons why we see the onset of mental health disorders appear in adolescence, is because we're going through that dynamic period of pruning and pruning these sort of pre-existing networks to refine them and make them more efficient. And so the experiences that we have during adolescence are really, really important, and they have more of an impact on our brain structure and function than in any other time of our life. So if we have kind of a tumultuous sort of adolescence and teenage years, and that can really affect the way that our brain prunes itself.
Very interesting. We've got another person who's wondering if there's a difference between male and female brains, presumably in the physical sense.
Yeah, there has been studies that have looked at how male and females process information. And so there is a slight difference in how we sort of, I guess, process information and how we deal with complex problems and tasks. They're not sort of saying, oh, men or women are better at some things than others, but there are definitely differences in the sort of functional connectivity of the brain. But that doesn't sort of mean that, men and women are better at doing certain things than others. It's just that we go about processing information in a slightly different way.
We are just past 8:30, and I will be wrapping things up after just 2 more questions. I think related to one of our very first questions, actually, neuroplasticity, is it affected in terms of how long it can go on for, or how well it happens after having a stroke?
That's a really good question. The answer is, that is a tough one. I mean, I guess it obviously, you can have neuroplasticity throughout your life, even after a stroke. And I've actually seen instances where people who've had a stroke come back into hospital and they've done really, really well with their rehabilitation. And talking to them, you wouldn't actually know that they had a stroke, and then we'll get them in the scanner, and the actual extent of their injury because of their stroke is extensive. And it's purely because their brain has managed to rewire itself that they can still carry out day-to-day functions. It was quite incredible, actually, this particular individual that I'm thinking about. So yeah, the brain does have an amazing ability to rewire itself at any age. Obviously, that ability does decrease slightly as we get older. So when I was working with premature babies, we obviously would see quite a few sad stories where there would be infants who would have huge strokes and ischemic infarcts, things like that. And their brain's ability to rewire itself was just incredible, because they're sort of brand new. So their brain's already primed to be growing and developing all of these connections. So it just sort of carries on and rewires itself. So I think it depends a lot on where the stroke is, what stage of your life you're in, and also a lot to do with your mindset. So particularly in the older patients that I saw in Sydney, it really did seem to be correlated with their mindset as to the improvements that they saw with their rehabilitation treatment.
Very good. Well, I've got a question here from Michael, I think it's a good one to end with. What have you learned in your neuroimaging research that has affected the way you try to lead your life, and how is that? How has it affected how you live your life? Yeah,
that's a good question. I am a bit of a worrier, and I get really anxious all the time, and I have problems sleeping just because I've always had problems sleeping even when I was a kid. And I think that understanding that loop in your brain and the impact of stress on your brain has really helped me to realize that actually most things aren't worth worrying about. And it was just almost a simple decision of, well, actually, I'm not going to let this, whatever this is, affect my brain and have long-term damaging effects to my brain. And that simple decision was quite liberating, actually. But I think it was only until I understood how my brain worked and how it likes to work that I was able to make that decision and actually worry about things a lot less.
Very good. Well, we are going to stop there. I'm very sorry if we haven't got round to your question, but we are going to stop so people can carry on with their evenings. And I need to say thank you once again, Dr. Kathryn Broadhouse, for a very entertaining hour. And thank you again to everybody who's chosen to join us and engage with the festival tonight and this week. And as I said at the beginning, there are more features that you might like to experience on the website, both coming up and in the archive. And that's at darwin.originalshrewsbury.co.uk. So with that, I'll bid you a good weekend and good night.